read the paper. It's a beautiful article. And so please welcome Dr. Thoru Pederson as he leads us through his thoughts regarding the 4D genome in Vivo, first steps towards a CRISPR toolkit. Thank you so much for coming.
Thanks indeed. It's a real honor to be here. This is an extraordinary thing that happens in your career when you get to be 73. You realize that there are some very wonderful places in the world of academic science and medicine that you've never been to. And I am ashamed to say that this is my first visit to the University of Michigan, and I hope it will not be my last, because I've been having just such a great day. I'd like to make two caveats. The first is that the host department is one relating to bioinformatics. I can tell you this is a subject of which I know absolutely nothing. And therefore, if any of you have questions about bioinformatics that's behind this work, I hope you will forgive me to defer them to emails from members of my laboratory that could flood in like an avalanche at midnight when I get home. The other is that this is the first seminar on this work that we have presented. We presented this a month ago at Cold Spring Harbor Laboratory. So this is very much a progress report, and this does not have perhaps the final formalism of a Cinderella biology story that you might have hoped for. So the functional organization of the cell nucleus and the folded state of the genome and its dynamics in space and time is actually a long-standing problem in the field of cell biology and gene expression, and therefore molecular biology. It's just been re-christened with a name, the 4D nucleome. We didn't do that. NIH did that through an initiative that was announced a few weeks ago. It simply means that there's a desire on the part of your former colleague, Francis Collins, to get those of us who are slightly shop-worn cell biologists of the nucleus to get really engaged in mapping out at some granular detail its dynamic functions as a tapestry of genome loci and their dynamics as a function of space and time. So in that regard, we find ourselves very happy to have seen that RFP, because we've been working on this now for a number of years, trying to localize genes in live cells. You all know that it's possible to fix cells and carry out nucleic acid hybridization in situ in a method that was developed by Joseph Gall at Yale University in 1968 and subsequently refined by others. That's a remarkable technique and one that has given us a great deal of insight into the static situation in the cell nucleus with regard to the location of various genes, or also the location of various RNAs, because one can also do RNA fish as well as DNA fish. 3 years ago in my laboratory, we decided that although we were working on many other projects that were going quite well, we would like to think about doing gene localization in live cells. And Han-Wei Ma in my laboratory, and I apologize for not having an acknowledgement slide, but this is the first seminar I've given on this, and I forgot to make one. But Han-Wei Ma is the second, is one of the two, not second, one of the two most talented postdocs of the 55 that I've had. And he has innovated this work to such a degree that I am not even allowing myself to be named on the patent that is being filed by my institution. He really innovated this completely on his own. This work has also been supported strongly by another postdoc in my laboratory, Pablo Reyes Gutierrez, and has benefited from an extraordinary bioinformatics collaboration at the University of Central Florida, laboratory of Xiaozhi Zhang, and another collaborator at the University of Massachusetts Medical School, Scott Wolf. The first part of this talk is going to be on work that has been published, and that deals with the use of transcription activator-like effectors, or tails, to label chromosomal loci in live cells. You've probably all heard of talins, which are tails that have a nuclease attached to them and have been used in gene editing. And actually, talins were the second chapter of the gene editing field, the first being zinc finger nucleases. However, if you take the nuclease domain off of a tail, and I'll tell you in a moment what a tail is, you can create a molecule that will label sequences of DNA. Tails are transcription activator-like elements in the sense that they are designed by protein chemistry to resemble highly specific DNA binding recognition sites and transcriptional activator factors. They have evolved in evolution to read specific DNA sequences in the double helix. And in fact, one of the reasons that we were very encouraged that we could use tails to label DNA sequences in live cells is because there is no need for denaturation of the DNA as there is in fixed cell fluorescence in situ hybridization. These tails are comprised of repeating arrays of amino acids, sometimes 13 to 14 amino acids in a row that are then repeated over a couple of times that bind specific sequences. And in this starting slide of the seminar, we show a tail that's been designed to recognize the telomeric repeat at the end of human chromosomes. The tail can be designed as an amino acid sequence that repeats on either to read this side of the strand in the minor groove or this side of the telomeric repeat in the major groove. And then, of course, each of these can be labeled with your favorite color. These are expressed as plasmids. All the plasmids and all the constructs that I'm going to show you here are in AdGene and are available. We've had over 300 requests in the few months since this paper was published. And I guess for the talk, we should put the lights kind of like halfway down. Halfway down means A.A. Milne, what was he say? Halfway down the stairs is a stair where I sit. It's not at the bottom. It's not at the top, but it is the stair where I always stop. I think that was Christopher Robin. But there is a luminosity level, which is just gracefully positioned so that it allows the speaker to see those who are asleep and does not yet further induce sleep in others who are not asleep. So after expression for 12 to 20 hours in human U2 osteosarcoma cells, one can see that the tail labeling of discrete sites, either in the Venus color or the cherry color, and here's the overlay. This doesn't say very much to you. He's lighting up some spots. The question becomes, is there specificity? So in this experiment, we have combined the tail live cell labeling in the upper row with post live cell labeling with immunostaining in the middle row. So these are live cells that are being imaged with the telomere tail. And this is cells that have been fixed and stained with an antibody to a shelterin protein that sits on the telomeric repeat called TRIF2. And this is an experiment in which the cell has been labeled by the tail in the live state and then fixed and imaged to show you that the tail that we designed to bind to telomeres lights up foci that are entirely coincident with the sites of immunostaining for the telomere-specific protein. I think for some of this, we might even want a little less light. I say that at my own risk. Thank you, Alex. So it is also possible to carry out other tail designs. So here in the upper row is a tail that we designed for not the centromere. The centromere is an ugly beast in the human genome. In fact, it was one of the many repeated sequences that was never in the original human sequence draft. With a tail designed to recognize a pericentromeric region. So we call this a pan-centromeric probe because it's recognizing DNA sequences adjacent to the centromeres of all the chromosomes. And here is in M. cherry red, a tail designed to a telomere. This gets a little bit tongue-twisting. This is a tail for a telomere. And you can see that there is a very interesting set of patterns. These, of course, do not overlap. They shouldn't, because the centromere is in some internal site in each chromosome, and the telomeres are at the ends. So that makes sense. In fact, if you think about it, you actually can use this as a yardstick to measure certain distances of interest. More compellingly, we happen to be able to generate through a collaboration with Karen Niga in the UC Santa Cruz Genome Center in the Hauser group, tail probes that were specific for pericentromeric sequences on chromosomes 18 or 15. So in these two cases, this tail is binding only to the centromeres of chromosome 18. And of course, the reason you see more than two is because this is perhaps a G2 cell, but also this is an aneuploid cell. This is a similar one that we designed for a pericentromeric sequence around the centromeres on chromosome 15, along with the telomere probes. Here, one can measure the distance between a specific chromosome centromere and the telomeres. And this begins to give us a yardstick for assessing how the many, many megabases of DNA on the linear physical map of a particular chromosome are actually conformed into a compressed distance in the intact live cell. Again, remember, this is all live cells, nothing dead. These are human U2OS cells, the same ones we had before. Yes, and we haven't done the confocal imaging in this slide, but we have done it. We can count up, and now there's always the possibility that in one particular chromosome, this sequence is not available to a tail. I was very dogmatic when I said tails can bind double-stranded DNA. That's absolutely true. But there is the possibility that certain sequences are covered. There might even have been regions in those telomeres that are covered. Now, and things get a little clearer when we go to diploid cells. This is just to show you, and I would just ask you to remember this when I get to the CRISPR part of the story, the CRISPR part, is that you can do these tail labelings in any color you want. There are now 18 fluorescent proteins. The spectral bandwidth is extremely good. The microscopy companies have not caught up. If any of you do this, you know that you cannot get filter sets that will filter discriminately between blue and teal, for example. It's very, very hard to do. And so what we do is we simply, we call this one, we call, yeah, Alex, you were right. This is really, takes some practice. I got it now. We call this teal because this fluorescent protein is teal fluorescent protein. This is cyan, but we image them in the same channel, and that's sort of how they look up. So we can make a polychromatic palette of colors and do multi-labeling, multicolor labeling. I keep walking over here thinking I have to advance the slide over there. Now, to get more specific, one of the things that we got very interested in is when we got these telomere tails was looking at cells that have different lengths of telomeres. You guys all know there's a whole gestalt about telomeres and cancer. It's a dogma that I think has been a little bit overdone, frankly. But anyway, in this experiment, what we've done is we've used a variety of cells. These are the human U2OS cells that I've been talking about. They are highly aneuploid. Here are HeLa cells, also highly aneuploid. Two different strains. Telomere length in these cells is about 70 kilobases. In these cells is about 10 kilobases. In these HeLa cells is about 30 kilobases. Here's a diploid cell line, IMR90s, in which the telomeres are very much shorter, about 3 to 5. And this is human retinal pigmented epithelial cell line, in which the telomeres are extremely short, maybe 1 to 2 kilobases. And while we do not claim to have a quantitative assay for telomere length, as you have with a molecular assay such as TRAP assays, where you use the reverse transcriptase activity of telomerase to make a product, there is a very nice correlation between the known telomere average lengths in these cell lines and the brightness of the signals that we see. We decided that it would be possible to perhaps do a tricky experiment of mixing those two HeLa cell lines. So we labeled one of those HeLa cell lines with a DNA dye that is blue, DAPI, and we labeled the other HeLa cell line that has the longer telomeres with a DNA-specific dye that is red. And then we mixed them together and we imaged them, and that's shown in the bottom. But what is more interesting is, and again, remember, these are live cells, is you can go to your FACS center. And when you go to your FACS center, and in our institution, and I'm sure you do here, we have FACS instruments that sort live cells. And not only, this is to me unbelievable, they not only image live cells and sort them, but they actually they give you a recorded image. Now those cells, as they fly through that aperture, are round. They've been trypsinized off the cover glasses or the plates of the cells. And so you don't have the high quality microscopic resolution of the nucleus and the chromatin that you have in the conventional experiments in our laboratory. And yet you are able to extract a lot of information even from those round cell images. I'm not going to go through this in detail. It's much too complicated. I really want to get to the new unpublished work. But you obviously get a number of pieces of information out of this. First of all, with either the DAPI or the red dye, you get cell cycle information. That's standard. But secondly, with the tail-specific probes labeled in green or teal, you get binning of populations that you can then extract out. And you can see that there is heterogeneity. And I haven't been able to convince you because I didn't set this up right. But this analysis is being done on a mixed population of those two helix-cell lines that carry different telomere lengths, each of which is labeled with a DNA dye that's either red, either blue or red. I think you can see when I said Han-Wei Ma is a very creative investigator. When he said he was going to mix the cells, I said, oh, no, you're not supposed to mix cells. That's not good. I mean, that's the low subterranean reptilian level of my intelligence relative to where he was thinking, which is way up there. And this is just the kind of image data that you get. I really don't want to go through this. But we have been now collaborating with some of our oncology colleagues who are extremely excited by this because it turns out, and I'll maybe just take a moment here to turn the slide off and say that all of that work is live cells, transient expression of these various plasmids for tails. Han-Wei Ma one day came in the office and he said, you know, you don't have to denature the DNA, do you? And I said, yeah, well, why are you telling me that? Because that's why you did this. And he said, but you know, I just made some recombinant tails in E. coli as proteins with fluorescent proteins. And what he had done, this sneaky guy, he had fixed some cells and he made up a buffer and he put his recombinant tail fluorescent protein in the buffer and just let it sit on the cells and brought me pictures that were as glorious as what you see there. Now you might say, well, why are you now reverting to that day of yesteryear, to use a Lone Ranger TV show thing? Why are you going back to fixed cells? Well, it took Han-Wei Ma a couple of days to make the fluorescent tail in E. coli as a recombinant protein. It took him 10 minutes to do the in situ labeling. In situ nucleic acid hybridization minimally takes 24 hours. Therefore, we now believe, and we've been working on this with some of our oncologists, particularly our hem-onc people, that it would be possible to take a sample of cells, a fine needle biopsy or a suspension of blood cells, and within 5 minutes, give a clinician a report on the number of copies of chromosome 9 or the length of telomeres and so forth, based on a fixed cell assay that resembles in situ nucleic acid hybridization, but is based on this technology. So there's a lot of new things that we're thinking of doing. This keyboard has W and D in different places. There we go. There's nothing because the cell is dead. The cell is dead. You're doing in situ hybridization, but you're doing it under a condition where you don't have to denature the DNA because the tail never wants to bind single-stranded DNA. It can't bind single-stranded DNA. This is part of the patent that we filed a year ago, and of course, it's been only a year, so of course we haven't heard anything from the patent office. Now, I want to, and then of course you can, our FACS Center also gives us data like this, where they send this back and they even tell us what is the z-axis looking like in the cells that are flying through. I mean, the laser beam is oriented in such a way that it's getting a whole cell image, but they can computationally extract out this kind of information. And you can also, again, remember, after the FACS is sorted and you've got the data on the heterogeneity of telomere lengths, now you've still got the cells. They're still alive. They've come out the other end of the fax machine, and they're still alive. Put them in culture and take a look at what they do. And you can follow them through several cell cycles, and I'm not going to go through those data. So that's where we are. Now, about 4 months ago, Hanwei Ma again came to my office. He comes to my office, and he says, I have a new project. I thought he meant he had a new project to propose. No, he sits down with his laptop and tells me that he has already done about 1/3 of the work I'm going to tell you about today. Though this is work that's been done by him in the last 4 months or so. So CRISPR. This is a bacterial immunity system that was discovered over a decade ago, in which there is an enzyme that is an endonuclease, Cas9, that is responsible for excising from the bacterial genome invasive DNA. It does so in the most ingenious way you could possibly imagine. It actually uses RNA transcripts made by the invading invasive DNA as a way to guide this pre-existing endonuclease to the very places where the invasive palindromic repeats have been inserted. This is called, the CRISPR stands for clustered regularly spaced palindromic repeats. So it was not clear 10 years ago that this system was anything other than an esoteric sideshow in the world of biology. And I remember that several people in our RNA group, Craig Mello and Victor Ambrose and the rest of us, started to teach this paper in our lectures. And as we were teaching, we thought, boy, if this could be repurposed, it would really be cool. Well, of course, one didn't have to wait long, because the CRISPR system has been repurposed and is now the major tool for gene editing. It is an explosively dynamic field. It's also, I must tell you, a very competitive field. And so what Hanwei has done is used CRISPR technology to label chromosomes in the same way as the tails, but there are some additional twists that I'd like to share with you. And this is where we are as of right now. As of right now. Here. I'm going to tell you a little bit about how we're using CRISPR to do multicolor labeling of genomic loci in live cells. I'm going to tell you about how we've been using it to look at disease-related repeat expansion loci in a number of disease situations. I'm going to tell you without data about how you can use CRISPR to stall RNA polymerase on an expanded allele in the disease myotonic dystrophy and cause an accumulation of toxic RNA nuclear foci to disappear. I should say, by the way, that if you think about it, CRISPR is RNA interference. It's not RNAi because it's not triggered by double-stranded RNA. However, it is an interference mechanism that is based on the sequence specificity of the small guide RNA transcripts going back to recognize their birthplace in the DNA. And therefore, it is RNA-directed interference. The big difference between RNAi and CRISPR is that RNAi operates at the translational level and the CRISPR operates at the transcriptional level. And incidentally, speaking of RNA interference, I think there's no one I know who doubts that there will be a Nobel for CRISPR, and it probably will come as fast as Nobels ever do. Alfred Nobel, wanted them all to be within 1 year, and that's not often possible, but the CRISPR Nobel will be very quick, I can assure you. And then most excitingly is our work on mapping individual chromosome territories. With Xiaoji Zhang, we have been able to go through now all the repeated sequences in the human genome that were not well characterized in the draft sequence and that have now been mined by him and others. And we have found sequences of repeats in every one of the chromosomes that is unique to that particular chromosome. And we have now been able to further refine those sequences to CRISPR enablement. And then finally, a little bit about barcoding. So the first thing is multicolor. So the Cas nuclease can be created as a GFP fusion, or it can be fused with any other color you want. In the experiments, I don't know why the C terminus is way over there. This is the C terminus over here. I don't know why it went over there. In the labeling Now remember, this is a system that most people are using to edit DNA. We don't want to cut the DNA. So in all the labeling studies, we use a catalytically inactive mutant of Cas9 that has no nuclease activity. So you'll see the term dCas. There are 3 well-characterized bacterial species that have CRISPR systems, and they each have slightly different DNA sequence recognition motifs called PAMs. I'm not going to go through this in detail, but simply to let you know that there are these 3 species with these 3 distinctive PAM recognition sequences that are related to the way the guide RNA attracts the attention of the dCas9. And so what we have done, we, meaning Hanwi and Pablo, is we have designed plasmids that are expressed using the U6 promoter to create the guide RNAs. The U6 promoter, I'm very happy to say, and very sad to say, was discovered in my laboratory by Gary Kunkel in 1986. I'm happy to say because it was a very important thing that Gary did. I'm sad to say because had we filed a patent, had we envisioned that the U6 promoter, U6 is a small nuclear RNA, if we had envisioned ahead of time that the U6 promoter could be used in trans to fire the transcription by Pol III of almost any small RNA you want and have correct termination, I would not be here. I would be out on my 150-foot yacht. But we never thought of that because there was no reason to think of it. And then in parallel, you co-transfect in any one of the 3 bacterial orthologs of the Cas9, the dCas9, catalytically inactive. You give it an inducible promoter, you give it a nuclear localization signal, and you give it the color that you want. And so, though this is a slide that would have been somewhat unbelievable if he had shown it to me without any data, the fact is this works. So we've been able to make some progress. So here's a simple experiment in which cells, these are U2S cells again, have been allowed to express any one of the 3 orthologs of dCas9 labeled in red, green, or blue fluorescent protein, along with the cognate guide RNA. And as you can see, we get labeling of loci in all 3. I'm not even telling you what the particular DNA sequence is that we're targeting here. It's irrelevant. This is just sort of a demonstration of the fact that we can do multicolor. Here's an experiment in which we have deliberately used guide RNAs designed to recognize the telomeric repeat. I want to emphasize the dCas9 system in any one of these 3 bacterial species is fairly discriminating about what sequences it will collaborate with as regards the guide RNAs. However, you can fool it in certain ways by mutating the dCas itself or by creating faux PAM sequences. Faux PAM sequences that confuse the bacterial Cas9 endonuclease to thinking that it wants to work with this small guide RNA that it normally wouldn't want to work to. This becomes relevant when you look at certain repeat sequences that are all purine, for example, like a GGGGG repeat that I'll tell you about in a moment. So what you can simply see here is that expression of these 2 Cas9 systems allows you, allows you to see telomeres in either color as you would predict. And here's the merged image over here, and the co-alignment is very, very good. It's actually quite a remarkable experiment when you think about it, because on these human telomeres, 2 proteins of bacterial origin that those telomeres had never seen before are being bound by those endonucleases. Here's just another example using different colors. Now, it occurred to us that telomeres has been a platform for some of this work just to demonstrate feasibility. But we're really interested in the whole genome. And so we began to, in collaboration with Xiaoji Zhang at the University of Central Florida, look for repeated sequences that would be of 2 criteria. One, that it would be a repeated sequence that would be present only on 1 chromosome. perhaps at many different places, and perhaps at many different places in many different copy numbers, but only present on one chromosome. And we have now found such sequences for all 23 human chromosomes, including the Y, which is the one that is so pathetically devoid of any intelligence or anything else. Second, we would want to eventually have a lot of these on any given chromosome. We would want to have 10 families of repeats, 1 through 10, all of which are restricted to a particular chromosome, but are located at many different locations. And I'll come to that. So the first one we chose was a sequence on two sequences on chromosome 9. One of which, what did I do? Thank you. One of which is a sequence that is on the long arm of chromosome 9, but just beyond the centromere. C91 we call it. The other of which is not labeled here is a sequence C92 that is at the distal end of this arm in the subtelomeric region. We designed Cas9 for both of them, labeled either red or green. I realize this is confusing, red and green, and express the cognate small guide RNAs in the same cells. These are retinal pigmented epithelial cells, diploid. So what you can see is that with M cherry, the red, we see 2 spots. And with the C91 in green, we see 2 spots. Now, these are not the same cells. These are 2 different cells. Well, that's good. So we got 2 copies, 2 copies. These must be cells in G1. Here's an example in which we have C91 and C92 in the same cell. Remarkably, we label both and know immediately that this microscopic spatial resolution is the distance of the live cell chromosome that reflects this distance of DNA sequence on the physical map. So we were quite excited by this, but it was just the beginning. Then we decided to push this a little further. This is just another example of this in 2 other colors. Now, what we decided to do was then push down the resolution. So instead of, in this case, taking chromosome 9, we took chromosome 13. And instead of taking a sequence down here, we took a sequence way up here. This is a repeated sequence. This is unique to chromosome 13, and one array of which is located here. Actually, this one is only located here, and is this close to the end of the chromosome, where you have the telomere. Designed the Cas9 systems for the C13-1 in red and the telomere in green, and we get labeling of both. And now, as predicted, they are much closer together. This is the microscopic readout of the physical map. Now, of course, in the cell, the physical map is not a linear piece of DNA. In fact, we do not know exactly how the DNA is conformed at the long arm of this chromosome. The telomeric repeat itself does not wrap around nucleosomes in quite the same way. But nonetheless, we're quite struck by these results. Now, in terms of disease, we've been able to do some initial experiments. Here's an experiment with myotonic dystrophy-derived cells. In myotonic dystrophy, there is a repeat of CTGs in a 3' untranslated region of one allele of this inherited form of the disease in a gene that codes for a protein kinase called DMK1. We designed a Cas9 small guide RNA for this repeat expansion and used it in vivo in live cells from a normal subject, as well as from a patient who was a boy who had the normal length of the repeat is about 60. This boy, who subsequently, of course, was deceased, this is a lethal disease, had 1,200 copies. I want to emphasize that up till now, we really are not sure of how many copies of Cas9 fluorescent protein we need to have on any given repeat to get a signal. But we can say from our tail work that we learned during that phase of this project that we probably need to have about 200. Therefore, it is not likely we will see the wild type allele in this particular situation, but that doesn't mean that we haven't got some Cas9 fluorescent protein on it. It's just not bright enough to be seen in the microscope. So here's the result for the normal fibroblast. And as you see, there is just a very low haze of green fluorescence. So this is very compatible with what I just said. We wouldn't expect to see 60 copies of the repeat. However, in the patient-derived fibroblasts, we see a single spot, which is the mutant allele. It must be the mutant allele because there's no other genomic DNA sequence basis for seeing a signal in this cell other than the expanded allele. So this gave us confidence that if we had a repeat that was at least this long, we could see it. And as I told you, I think we now are down to an estimate of about 150 to 200 repeats that we could be able to see. In this work, we have done another side project on the DM that I don't show data for. It is the fact that using dCas9, we can stall RNA polymerase as it runs along both the mutant and wild type allele. The stalling of RNA polymerase on the mutant and wild type allele allows us to do FISH for foci of nuclear RNA transcripts from the mutant allele that are toxic. These transcripts have such a long repeat of CTG that they form coagulated foci in the nucleus. In those foci, they suck up so much of the mRNA splicing factors that you end up with a global screwing up of splicing of all transcripts. And this is thought to be the pathogenic basis of this particular form of myotonic dystrophy. You have a generic inhibition of mRNA splicing because you've created this huge stockpile of these aberrant transcripts. After we stall Pol2 transcription with Cas9, we find that those nuclear RNA foci disappear in the great majority of the cells. And this is the basis of a March of Dimes grant application that we were just invited to submit. Now, remember, this is in the 3' UTR. It is quite possible you can also stall transcription of the wild type allele and get a functional protein. And so there is a therapeutic interest in this possibility that the March of Dimes is quite excited about. March of Dimes is quite ironic because Gil and I were talking about the fact that Jonas Salk chose your medical school to make his announcement after the trial because Tom Francis was here. So a little bit of polio connection there. Now, here's I'm almost, hopefully I'm almost finished. Yes, I am almost finished. Here's another example. So this is ALS and frontal temporal dementia. Here, we have a different kind of repeat, and this goes to the limitations of Cas technology that I was mentioning before. Cas does not like G. Doesn't like it at all. However, there are manipulations that Scott Wolf, our collaborator, has helped us do that have been able to get to the point where we can begin to attack this repeat. So in ALS, there is a gene called C9orf72. And C9, ORF standing for open reading frame, is now thought to be, by the neurologists, the most determinative gene mutation for inherited ALS. It also, ironically, shows up in a lot of sporadic ALS, which is kind of mysterious. I'm not sure I really understand that. So we have used this Cas approach to attack the GGG-GCC repeat expansion in human fibroblasts. And using our technology, we've been able to identify single nuclear foci of DNA labeling in any one of several constructions, only two of which I'm showing you here. Now, this doesn't get us very far, but it's very, very encouraging. And we've also now recently gotten results for this for FTD as well. Oh, I just did again what I wasn't supposed to do. Now, I want to finish by getting into the real holy grail, which is mapping chromosomes all together. And the first thing we thought we would do, we better check and find out if our live cell CRISPR labeling method recapitulates gene localizations and indeed chromosome localizations that have been recorded before in fixed cells by fish. And one of the best examples of this is the dichotomy between human chromosomes 19 and 18. Chromosome 18 is a sad, gene-poor chromosome, and chromosome 19 is a happy, gene-rich chromosome. And Wendy Bickmore and other laboratories had shown some years ago that this difference in gene density is correlated with nuclear location. And in particular, that the relatively gene-poor, not gene-inactive, but gene-poor, there's plenty of transcription, chromosome 18 was situated at the nuclear periphery, and that the more active gene-rich chromosome 19 was located in the nuclear interior. So we designed the appropriate Cas9 small guide RNA system, and we were able to show very clearly that we were, recapitulating the result. Here's chromosome 19 labeled with dCas9 and a green fluorescent protein tag, and the suitable small guide RNA in a more interior location. And here's chromosome 18 plastered up against the nuclear envelope in association probably with the nuclear lamina. But that's only one example. We've also now been able to go on and look at this with respect to chromosome 3 and chromosome 7. And it's very interesting that we've anticipated the literature here, because chromosome 7 in these experiments is like chromosome 18. It's plastered right up against the nuclear envelope. And we didn't know whether this was a positive control or a negative control. We don't know it. And now we've learned in the last few weeks that actually chromosome 7 has this localization by fish, although for our publication, we're going to have to do that ourselves, since it's not appropriate to say personal communication of unpublished data not shown. And here's the combined images of 3 and 7. So we're quite excited about this. And then the last thing I would like, then quickly, let me say that another mapping we've been doing goes back to our long-standing interest in the nucleolus, a part of the nucleus that arises from the genes for ribosomal RNA that are clustered in tandem arrays on 5 of the human chromosomes, the acrocentrics, and 13, 14, 15, 21, and 22. And what we have been able to do is design the appropriate Cas9 small guide RNA probes for the rDNA repeats and begin to look at their location. Now, you might say, why are you even bothering? The rDNA repeats, you just said, are the basis of the nucleoli. I mean, the nucleoli are a cytological manifestation of the rDNA repeats in their transcription and the assembly of nascent ribosomes. They're called nuclear organizing regions for that reason. Turns out only half of the rDNA that we can detect by our Cas9 probes has a Nucleolus vicinal localization, shown in the red arrows. Nucleolus propinquity. In the white arrows, it's not near the nucleoli. And it turns out that this is probably a manifestation in these live cell images of the fact that there is silencing of a lot of the rDNA in human cells. And in fact, there is an indication that there may even be some clinical significance to these observations in various patients. So we're very excited about that. And then finally, let me just finish by saying that Xiaoji Zhang and his student, Andolin Nazari, have now completed their complete bioinformatics mining endeavor of chromosome 9. And they have focused on the long arm of chromosome 9, and they have now been able to define a large number of repeated sequences which lie in that long arm and are not present in the short arm, nor are they present in any one of the other 22 human chromosomes. They are arranged in a non-periodic array. Why would they be periodic? They're arranged in that way. We do not know whether each and every one of those repeats have enough copies that we could pick them up by Cas9. Some of them might have 10 repeats, some might have 50. Those will miss. But we already know that many of them have 200 or more. And what is more, the blue ones are all the same, but the red one is different. And so we figure that if we now design at least several dCas9s for these repeats, we'll be able to light up a large region of the chromosome. And now Xiaozhi has begun to find a second family of repeats that lie all throughout, that are not as many as these, maybe about 12. They are lying all throughout, and they are a different sequence family. So we will have at least 2 colors for barcoding this chromosome arm. And of course, with 17 fluorescent proteins, and our microscope can't simultaneously resolve 17 colors, but we think it should be possible in the near future in this very fast-moving field to be able to do barcoding of chromosomes in live cells. This means we can look at translocations not only as they happen in the father or the mother, we can look at translocations as they are actually happening in a laboratory environment in cultured cells. So with that, I will simply say that this is our current work, localization and tracking of dynamics. I have not had time to talk about the fact that we have a switchable system. If you think about it, if you're trying to optimize your CRISPR editing of a gene, you first need to find the conditions where you're getting the maximum CRISPR binding. Our system allows that by using the dCas9 mutant that cannot cut. And so we have a platform that we've designed in which large numbers of cells can be analyzed for the efficiency of dCas9 labeling of a targeted gene. Those are then sorted, and the cells that are brightest are taken back and are subjected to a second round of a switched dCas that is catalytically active to accomplish cleavage, not of the same sequence, because don't forget, it's already got the dCas9 stuck to it, but to marker sequences that border it that we can excise out. This has high throughput potential and very fast turnaround, and we're very excited about it. So I thank you for your attention.
Let's get some good questions going. Dave's ready.
Okay, so Thoru, I've got a short and a long question. The first is, where on the ribosomal gene repeats are your probes relative to the transcription unit?
They are in the coding region.
So
They are in the transcribe region.
So by analogy, they could be in the path of the polymerases.
Oh, yes, that's true, and it's a good point. And they could be stalling the polymerase, and that could cause it to move away from nucleoli. That's an excellent point.
Okay, so the related long question is that you always worry that when you put 200 copies of anything down on a chromosome, it might change the behavior. But you've got a really interesting system with which to block transcription. As you're seeing it in the ALS examples, and ask, does that affect the behavior of the locus by an independent means? Have you started looking at that at all?
No, I mean, the collaborative team there has suggested that as well. It's a very good point, and I think that it's something we really have to do. There's different predictions you could make, but right now we're just trying to quantify the degree to which we've stalled the polymerase by doing RNA-seq. And we're doing CHIP also to make sure that we've got the polymerase stalled at the right place. I mean, it should be coming up to that point and then stopping. We shouldn't see transcripts beyond it and so forth. So we're checking that out. That wasn't such a long question. Yes, sure.
So one of the questions in the organization that is of interest to many of us is what the variability is in the population of cells. And to be able to answer some of those questions, I guess the assay needs to be something that is robust throughout the cell population. So I'm curious about this assay in terms of, if you look at the whole cell population, what proportion of the cells show different numbers of foci?
Yeah, that's a very good point. Well, in the retinal pigmented epithelia, which I would say is our best system so far because it's diploid, we don't see number variation for any particular target. We see 2, 3, or 4, which is what exactly you should see. The target we're looking at probably isn't concurrently, the 2 alleles are probably not concurrently replicated, so that's why we see 3. But I think it's a very good point that we need to be careful about in general. As far as the spatial concordance. I can't speak to it yet because we haven't done three-dimensional coordination. What I, in other words, saying it's all 2D in a sense. You know, 18 is always at the nuclear periphery, 19 is always in the nuclear interior. But is 18 always at 3 o'clock, or is it sometimes at 7 o'clock? We haven't gotten to that geometry yet, but that's something that we need to do with
Let me ask
their z-axis focus.
if I may ask a follow-up. You showed us an example where you had mapped these repeat elements bioinformatically identified. I think it was chromosome 13 or was it 12? You saw only one of the chromosomes in those images. Do you have an idea why you're only seeing one chromosome?
No, we see two in some. I mean, there are cells where we see one and cells where we see two. And that one we may not have focused down far enough. The thing that really excited us was to see consistently only the expanded myotonic dystrophy allele. That gave us some confidence.
Hi, this may be impertinent, it's a little tangential, but it is about the myotonic dystrophy. You made the comment about, I couldn't tell whether it was your speculation or somebody else's speculation, that the deleterious effect might come from this generic jamming of the splice machinery, right? Yeah. If that were the case, then it raises, well, to my mind, it raises the following question. How did you see that boy, in the sense that that's such a generic mechanism that would seem to make a fetus unviable?
Yeah, well, I think, first of all, that is not my speculation. That's sort of a dogma that's in the myotonic dystrophy field. It's been there since David Hausman and other people were working on it back in the 90s. I think the answer must be that this is not all or none. I mean, after all, it's not embryonic lethal. In fact, it's not even early onset, relatively speaking. I mean, that boy died when he was 13. It's like some of the diseases now that are being considered to be translational slowdown. I mean, why would you think translational slowdown would hurt you ever? Because you'll make the protein sooner or later, but there's a number of pathologies now that are beginning to look that way. So yes, this patient had one of the longer repeats, and that's from the Coriell library. That's the one we chose to start with. But I think the idea is that you get mRNA processing botched up enough that either qualitatively in terms of alternative splicing or kinetically in terms of just mounting the proper steady state level of message, you've just created a kind of a bad situation. And myotonic dystrophy has upstream and downstream effects at the neuromuscular junction. There's a motor neuron component of the dystrophic muscle post neuromuscular junction as well. So you've got two different gene expression platforms also playing in this game.
Did that boy die young? As, you know,
13.
out of the well, you said that, but I mean out of the relatively out of the population.
I'm not an expert. I think that's a little on the later side. I think these boys. It's not like Duchenne, where I think the age of death is a little bit earlier. This is a little less severe than Duchenne, which is always seen in the sartorial muscles when the little boy just cannot push himself up, and that's when you know something's wrong. I think this was a little bit on the longer lived side. Sure. Yes, it's already been done, actually. About 6 months ago, no, let's see, what month is this? Is this October? It's October 1st, isn't it? Okay, yeah, it's October. Well, I think about 6 months ago, the group at UCSF, Jennifer Doudna, John Weissman, published a paper in Cell in which they were able to localize a single gene. However, they localized the single copy gene by virtue of the fact that in one of its introns, there's a repeat. And you could say we've done that for DMK1 as well, DMPK as well. I think it's a very good question, Michael. I think what I would say, by the way, I'm very pleased to say that I was the external member of Michael's PhD thesis research advisory committee when he was at Lehigh University, and I am so proud to see you here and doing all the great stuff you told me about at breakfast. I don't think we can go too much further in sensitivity. If you talk to optical engineers, they'll tell you the charge coupled device invention has the quantum efficiency that it has and that it can't have any more. Just can't. Now, I don't know why they say that, but that's physics. The brightness of the fluorescent proteins is what it is. Those have been tuned in evolution to create a spectral shift, in the case of GFP, from green light to blue, that is important for the mating of a jellyfish. So why are we just saying we can make it any brighter? I want it to be brighter. I want our cameras to be more sensitive, but I think we may be a little bit stuck. It's a good point. Actually, the jellyfish, it's blue light to green light. I said it wrong. Thank you.